Thank you very much. Thank you, Academy, for having us here. The name is a misnomer for my work, actually. So I am not involved in DNA fingerprinting or diagnostics. We are a basic science group at CDFD. So before I begin talking about our science, I should start thanking the funding bodies which have been or which had and have been supporting our work throughout these years. The work that I am going to present today is fully supported by the DBT India, DBT Welcome India Alliance Fellowship. So what I am going to talk about is some of I will give a glimpse of what we are doing in the lab for last 15 years. So our lab is interested in understanding the systems that control cellular homeostasis, and one such system that I am going to talk about is the phosphorylation dynamics that are being controlled by two different sets of enzymes called kinases and phosphatases. So as everybody knows, probably the kinases are the enzymes which transfer the phosphate group from ATP onto the substrates like proteins, lipids, or any of these substrates, make them to a phosphorylated substrate, and this phosphorylated substrates can be active or inactive depending on the context and depending on the cellular role. So this homeostasis of phosphorylation is being maintained by the set of enzymes called phosphatases, which can remove this phosphate group and make them unphosphorylated. So the dynamics between the kinases and phosphatases is very critical in the cell for any cell to survive, and this is a most studied modification in the cell where it is being attributed. You can think of any cellular process it is involved, right. So the interesting part is that the protein kinases are like favorites for every biologist in the world, and all the companies, all the biotech companies or pharma companies, they are being the most druggable targets in the history. However, the protein phosphatase have been ignored for a very long time and is still ignored in the field. So we took up on the challenge of studying the protein phosphatases in the lab, and to just give a glimpse of what these enzymes are. So there are nearly 210 phosphatases in human cells, and based on their substrates they act on, whether they remove phosphate on serines, threonines, or tyrosines, or both of them, or histidines, or even lipids. So they are classified into different classes, and they are also subclassified based on their activity. So the details are not very important. So what is important to know is there are very limited number of phosphatases in the cell of a human cell, which is around 210. So the questions that we are trying to ask is, and are relevant for the field, of many in the field. So what is the functional rapporteur of all the phosphatases in humans, and what are different functions? These phosphatases have been existent for a very long time, but the functions are not very well mapped. And the second question that we are trying to address is, how are very limited number of these enzymes are able to control the phosphorylation dynamics of the whole cell. So if you imagine a cell, so normally the cell has around 20,000 proteins roughly, and if you add up the number of isoforms for each protein, it comes around 400,000 proteins of all in the cell. However, there are only 210 phosphatases, and every protein in the cell is at any point of time, at some point of time, is known to get phosphorylated. So the question is, how is this very small number of phosphatases control nearly 400,000 proteins in the cell? So that's a question that is there remaining in the field, and there are some phosphatases which are known, but the mechanisms are not known. So we are trying to attribute new mechanisms for this, and one of the ways is that one of the ideas that we have in the lab is whether phosphatases do they require the catalytic function always for their function. So that's a question we are trying to address. So these are some of the studies that we try to make some answers for these questions. I'll give a glimpse of what these three questions are in three different stories. So the approach that we are taking in the lab is a very simple one. So we try to, so everybody thought that, I mean, these phosphatases used to work in isolation. They are like they act on one substrate on alone. So the reality is that they are not in isolation, they work in groups. So to understand the groups of proteins that are associated with these phosphatases, so what we did was to express these phosphatases in cells, each of them individually, and then do a simple biochemical purification to understand what proteins, what partners they bring along in the cell. So use a simple proteomic approach, a mass spec based approach, to identify the interactors. So the details are not so important. So what we try to do is, so there is a old saying that if you know your friends, I can tell you who you are. So we are trying to find the friends for these phosphatases, right. So this is the network that we built. It's a years, I mean, quite a few years of work. We try to build the network of all the phosphatases that are and the protein, the interactors of all the phosphatases. The yellow ones are the orange ones are the phosphatases, and the proteins which are in blue are the interactors. So this is a network of made from nearly 70,000 interactions that we found by using mass spec, and by crunching all the numbers by using different algorithms, we could get around 2,000 proteins which are high confident interactions for these phosphatases. What is interesting is that there are 95% of these interactions or friends that we found are new, so they are not known to exist in the literature so far. So the challenge is now to identify what these friends are doing with these phosphatases, or not only friends, they could be some enemies also. So the first question that I'll try to address is, can we fill the mechanism for missing phosphatase functions? So to address these questions, I need to take one example. I'll take an example of a phosphatase called P10. Most of you probably know P53 as a tumor suppressor. P10 is second most mutated gene in human cancers, right? It's a lipid phosphatase. What is known about this phosphatase is it's mutated in most of the cancers, and it is a negative regulator of growth factor signaling. So it has to stop cells dividing, right? It is a negative regulator of cell growth. So what normally happens is, when you have growth factors added in cells, they bind to receptors which are specific for them, and then mediate some of the phosphorylation events. The main phosphorylation event that happens is the conversion of lipid from one of the smaller lipid formation to the higher lipid, which is a three phosphate groups added to it, and this acts as a sensor for activating a lot of signaling pathways in the cell. The details of the signaling pathway is not important. What is important is that this ligand binding, the growth factors that bind to the receptor, make the cells activate the growth factor signaling, and the cells tend to grow and then proliferate and survive. What this P10 does is a very simple blockage of this conversion. So it reverses the conversion of the phosphate group into a non-phosphorylated form here. So thereby it blocks the all the signaling downstream. So you can imagine that if you have a defective protein of P10, a defective or a mutant protein, it leads to cancers because it leads to uncontrolled growth or uncontrolled proliferation of cells. So that's how it works as a tumor suppressor. And so it's not only the mutations that are important for regulation of this, lost in are important for the tumor suppression activity, but there are other mechanisms that have been found for this protein, where it's not mutation of the protein. The mutation can, there may not be any mutations, but still the protein can be regulated at protein level itself, right? So the levels of protein can also be regulated, where a process called ubiquitination, it's other than phosphorylation, this is a different modification where a protein is added to another protein. So essentially what it does is, it is a signal for making proteins to degrade. So that's very well known for ubiquitination. So this is identified as a regulatory mechanism for this protein phosphatase in cells, and this has been the enzymatic machineries, or the proteins which are necessary for this modification, is also identified. But the twist in the story came when one of our colleague made a mice model where we deleted the NET41, which is the enzyme which mediates the ubiquitination, right. So this deletion of this in the mice is not able to show any changes in the P10 levels or stability. So this is absolutely contradicting what is already known. So that's where we stepped in and then tried to address the controversy and see if there are enzymes or in the list of interactors that we have now, can we find any of them being substituting for this function of net4. So there are quite a few proteins which are found interacting with this phosphatase, and one of them turned out to be an E3 ligase, which is very similar to the one which is here, right. It's from the same family, but it is not related to it. So what we showed is that this is the physiologically relevant E3 ligase. So where it is the one, that is the one which is binding and not the one which is known, and that is able to mediate the transfer. So this is the classical way of testing whether this protein is modified or not. So you can see that this is modifying very efficiently, and it is physiologically relevant. Not only physiologically relevant, it is disease relevant, because when you make cells which are devoid of this gene and put them into mice which are immunocompromised, you can see that normally these mice can form tumors, but this one, the cells which are lacking this are defective in forming tumors. So it's an oncogene which is known to regulate this tumor suppressor now. So it's a well established oncogene now in the field. So I'll switch gears, and this is the example for setting up the mechanism for known phosphatase. So what I'll try to do is switch gears and then try to address the second point, where we try to identify new functions. Can we identify new functions for this? So the phosphatase remains the same. So for the simplicity's sake, I kept P10 as a phosphatase. Other than this, there are many unknown proteins which are there in the interactome. So we have not seen these interactions before in the literature. So the question is, all the functions of P10 has been shown to be dependent on its lipid phosphatase activity so far in the literature. So since this is binding to many proteins, so the question that we asked is whether it is acting as a protein phosphatase as well, in addition to lipid phosphatase. Of course, the answer is yes. So before going into that, I'll take this as an example. So RAP7 is a protein which we tried to see if that is a substrate, and this protein is very critical for a process called endocytosis. So as I said earlier, so growth factors bind and then mediate the signaling. So one of the ways I said is to dephosphorylate at the lipid membrane and then stop the activity of growth factors. The second mechanism that is known is this growth factor ligand, once it is bound, it is activated, the signal is shut by a process called endocytosis. It is taken up by the cell through a process called clathrin mediated endocytosis, and this goes through a smaller vesicles in the cell, and they travel through all out the cell depending on what their fate is, and they end up in a small vesicle called early endosome. So what they do is, this is the sorting station, right. So this is like a post office, right. So where you sort where each cargo has to be delivered. So this is where it has to make a decision, the cell has to make a decision whether to convert this into a late endosome mature and then fuse with the lysosome, which is the degradation machinery, right, and then the signal is stopped. However, so if this switching is not happening, it can go back and then start the cycle again, right. So it can signal the cell to proliferate again. So the critical protein in this whole setup is the RAP7, which decides the switching. If RAP7 does not exist in the cell, this keeps going. Okay, the switch has to be dependent. The RAP7 has to be the switch which decides the maturation. Right. If RAP7 is compromised, there is always uncontrolled proliferation of cells. So the question is, now we have a tumor suppressor phosphatase interacting with this. The question, what is it doing? So what we went on to show is that it is regulating the switch function of RAP7. So this is a simple assay where we show that we label the cells with the EGF, that is the growth factor ligand. So this is a fluorescent labeled ligand. You can track the movement of this ligand in the cells by using fluorescent microscopy, and you can see that this goes into early endosomes. The early endosome is the marker that is used as EA1. So you can see it enters early endosomes here very early, and then you can see that it is coming off the early endosomes. The green one is the E1 and EGF is the red one. So they start separating. So it moves into the late endosomes, right. So where there is RAP7 present. So whereas in P10 null cells or depleted cells, so the EGF which is going into early endosomes, remain in the early endosomes for a very long time. So you can see the yellow is the indicator of resident protein, resident TGF in EE1. So it continues to stay in the early endosome. It is not able to move into next step, right? So the reason for that is that it regulates the maturation of RAP7 from the inactive form to the active form. So when you have this phosphatase which dephosphorylates it, this is now a new substrate for P10 because it's a lipid phosphatase, as I told you earlier. Now it is a protein phosphatase which can remove these phosphates on proteins as well. So RAP7 it stays in an inactive form and it needs to be dephosphorylated to become active, and then goes into the endosome and then make the endosome mature. Okay. So that is how P10 regulates. This is a completely new function for P10 other than its lipid phosphatase activity earlier. So the third point that I will touch upon is, so as I said, there are limited number of enzymes. So how do these enzymes control the whole proteome? So the answer for that is very simple. So we try to hypothesize that it probably forms many different set of combination of enzymes. Yeah, this is probably the last slide. So where the catalytic subunit is same, okay, the enzyme is same, but it can have different regulators or adapters for it to form these holoenzyme like structures. These adapters can be very specific for different specific substrates. So the same enzyme can act on different substrates depending on the combination of the adapters, right? So that's what is known in the literature. There are only 7, this family of proteins which has 7 are known to form such kind of holoenzymes, and we had to there are remaining 200 plus enzymes. So we went on to show that it is not just restricted to only a small set of proteins here. So we now have evidence to show that this forms higher order complexes and these holoenzyme like complexes with other enzymes as well. So that is one of the examples where we see that this is a phosphatase which is of a different class. It is known to be in the nucleus where it acts on its nuclear substrates and does the functions which are relevant for this. What we went on to show is that this, when it is bound with a different factor like B15, it is a adapter, it gets into the cytoplasm and where it has access to new set of substrates in the cytoplasm. So this gives a totally different function for this enzyme, right. So the enzymes based on the location or based on the adaptability, I mean the interaction with its partners, can decide what kind of substrates it can talk to. So, so it has a new set of substrates here. So these are the 3 examples to understand 3 different points. So I think I'll stop there before I'll just summarize what we just discussed. We take home points are they have diverse unidentified functions in the cell. We need to find more of them. They may not necessarily require phosphatase activity. I didn't touch upon this point because lack of time. So they do not require catalytic activity all the time. So they are active without enzyme. They assemble multimery complexes to increase the diversity. So I'll stop there and then take any questions. Thank you.
Thank you very much.
What is it that you have by way of tissue specific expression in the system?
Yeah, so there are some phosphatases which are specific for some tissues, but many of them are ubiquitous. They are present in most of the tissues. So however, at the mRNA they are expressed in every tissue. So at the protein level they are regulated. Some tissues have higher expression and some don't have. So there are hematopoietic specific phosphatases, there are neurological phosphatases, but they do exist in all types of cells. Yeah.
Phosphatases have a DNA binding domain. They do bind to the DNA, and yes.
Yeah, you asked that question. Very interesting. It does. They do not have a DNA binding domain. So I didn't come across any phosphatase which has a DNA binding domain, but they do go to chromatin. So you had to. There is a new study from our lab which goes. There are protein phosphatases which go to chromatin and then regulate transcription processes and all. So it's binding to chromatin associated proteins. Yeah.
Sir, what are the software you're using to fetching your results, sir?
Sorry.
What are the software you have used to fetching your results, sir?
Which one? There are plenty of results. So
Yes,
the
sir.
one which is network is the cytoscape that we use.
Mapping and sort of stuff.
Yeah, mapping is done by cytoscape. The MassPic is done by different software. It is not in house, so it's done by Harvard facility. but all the networks are built by Cytoscape.
Okay. Thank you.